Hello, welcome to Medical Immunology. Today we will continue with the theme of mucosal immunity and ulcerative colitis by looking at a phase 2 clinical trial of etrasimod. This will be the basis for an upcoming quiz. My hope is that this paper will be much easier to read than the previous paper. Hopefully this is going to be kind of reinforcing a number of the basic concepts that we learned going into the original mucosal immune system lecture. So this is taken directly from that mucosal immunity lecture. But essentially, these immune system can help distinguish commensal microbiota from different harmful pathogens. So this is a major immune organ. There are so many immune cells within the intestinal system, and specifically within the colon. Normally, these are helping us sense good bugs versus bad bugs. But for some people, they develop colitis. And so this colitis could be triggered by a infection, and then that infection causes this cascade of chronic inflammation. Or it could be that people just have genetic susceptibility. A lot of times is that for certain people is that this colitis might run in the family, is that you might have a genetic predisposition where maybe there's mutations in your immunoregulatory genes. Maybe there's mutations in your pro-inflammatory genes that gives you a kind of hair trigger to induce pro-inflammatory immune responses in the colon. So this is ulcerative colitis. This is quite debilitating disease for a number of patients. So this can present in a number of different ways. So here, the most common form is proctitis. So this is inflammation, particularly in the rectum, and this can cause rectal bleeding. This could cause urgency, like you have to go very frequently. You could have left-sided colitis that involves the sigmoid and the descending colitis as well. And here you could have ulcers that could form in this region of the colon. So this could be proctitis plus diarrhea and abdominal cramping, which is very uncomfortable. Or you could have extensive colitis that lasts the entire colon. And this is, quite unpleasant. This is, you also have potential systemic symptoms like fever and fatigue that are quite debilitating. And so this will be diagnosed through a colonoscopy. So you would take a bowel prep to remove the feces, and then go in there with a colonoscope and check to see, what do you see? Can you take a number of different samples? Just visually, you can see these ulcers. But going through, you can take a number of samples, bring it in, and start really kind of, understanding the type of inflammation that's happening and where it's happening. And a lot of this will guide how aggressive you need to be with treatment. So we've gone through this in the previous lecture, but the goal of today's lecture is to really focus on these sphingosine 1 phosphate receptor modulators. So the idea is that there's a number of patients that are refractory to existing therapies on the market. There's an unmet clinical need in ulcerative colitis, where people with severe colitis continue to have disease, and this disease is not responsive to other therapies on the market. So therapies on the market that might work for some people include anti-integrin therapies, cytokine therapies, anti-signaling therapies like tofacitinib. But here we have a different mechanism, a new mechanism called S1P receptor modulation. So let's understand how these work and how these keep immune cells trapped in the lymph node so that they cannot make their way out into the bloodstream and then back into the tissues. So first, we have to understand clinical decision-making. When and where do you decide to provide what drug? Fortunately, the experts in the field, the key opinion leaders, the thought leaders in the field, the people who know these drugs the best and who have run the clinical trials for these drugs, they understand what is happening. And they've put together these beautiful workflow documents, this kind of algorithm that helps you know what to try when. So let's take a patient that has moderate to severe ulcerative colitis. And keep in mind, this workflow is a little out of date by a couple years. So there's been a couple drugs that have been approved recently that are not in this workflow diagram. But for the purposes of this class, this is going to be a very helpful diagram to just kind of walk through the clinical decision making. So you have moderate to severe ulcerative colitis. So this is active disease. So this is a relapse where you're having active inflammation. This is going to be diarrhea. It's diarrhea. There could be blood in that diarrhea. Could be quite uncomfortable, maybe painful, depending on this type of colitis it is, whether it's proctitis or extensive colitis. But let's say it's moderate to severe colitis. So pretty uncomfortable. So in this case, the first thing you want to do is you want to tap down that relapse. What is an effective immunosuppressive strategy, therapeutic strategy to tap down that inflammation very quickly? It's going to be steroids. So oral corticosteroids, in particular prednisone, you're going to give high doses of steroids, and you're going to see, does that work? Does the bloody diarrhea stop, or does it continue? So let's say it stops. Let's say the steroids were effective at stopping that inflammation. That's good. So this patient is responding to steroids. Great. Now we need to go to the next step of treatment. What is the next step of treatment? The next step of treatment would be to maintain that patient in remission. So this patient had relapse disease, active inflammatory processes. But now this patient is in remission. The steroids stop the inflammation. They are no longer having active disease. So we don't want to keep the patient on steroids chronically. Is that if we can avoid using steroids, because steroids are associated with adverse events, we want to try a different drug to maintain that remission. So one drug is oral or topical 5-ASA, 5-aminosylic acid. So this is just kind of a general antioxidant, very safe, and you can take it at very high doses. And what this can do is this can just act to be an antioxidant, help suppress some of those reactive oxygen species. And what that might do is that might keep this patient in remission. So if you 3 to 6 months later, you follow up the patient and they haven't had any active flare of disease, they haven't had a relapse. At that point, you continue therapy and reassess every 6 to 12 months. So that is the best outcome. But some clinicians, depending on the pathology results, some clinicians, depending on the patient's previous history with other drugs, they might want to try different approaches, maybe like an anti-metabolite like thiopurine, or go directly to a biologic like an anti-TNF or an anti-integrin therapy. In this case, if those work, you maintain those therapies and continue to reassess. But if they don't work, at that point, you start switching up. So here you would go from the small molecule 5-ASA, the safer molecule, now to the more expensive biologic that requires infusions, requires more therapeutic monitoring. And at this point, you would switch to the anti-TNF or vedolizumab. If they're on TNF and they don't respond to TNF therapy, at that point, you switch over to vedolizumab therapy. And so you have a couple different options. And so here you're just trying the different drugs, seeing what that works for that patient. Unfortunately, there's no good diagnostic biomarker to say, oh, this patient will respond to this drug, but not these other drugs. It's just a matter of trial and error and seeing what works for that patient. So let's say that patient over time does not respond to those steroids. So this is a patient that has active disease. That active disease could be, a relapse that you have down here for patients that were being previously tried on other drugs and then just relapse. You put them back on steroids and seeing if that taps it down again. But let's say it doesn't tap it down this time. So if you have a patient with moderate to severe ulcerative colitis and the steroids don't work, the patient continues to have this bloody diarrhea, this relapsed refractory disease, at that point, you need to get more aggressive. You need to do IV steroids, and the IV steroids, intravenous steroids, will really be a sledgehammer to the immune system. And if that works, at that point, you need to decide, do you go with thiopurines or the biologics like anti-TNF or anti-integrin? Or if that doesn't work, if the patient is just totally refractory to the steroids, which unfortunately is quite common, at that point, now you have to get aggressive with the biologic therapies. That's going to be vedolizumab. That's going to be your anti-TNF drugs. If they don't respond to IV steroids, hopefully these drugs will tap it down. And here you want to look, does this patient have remission at 3 to 6 months? If yes, then you continue your patient on these drugs. But if they don't, if they continue to have relapses, continue to have active disease, at that point, you want to start switching. So you switch from vedolizumab, or you change to anti-TNF, or you intensify the dose. You do a dose intensification, the vedolizumab DI, or you could change to another TNF drug, increase the dose, or change to vedolizumab. If that doesn't work, at this point, you want to consider surgery. And for this, you would remove the inflamed portion of the colon. Could be, for proctitis, it could be a smaller removal, or for extensive colitis, it could be full colectomy. Full colectomy and surgical removal of portions of the colon, these are expensive procedures. These are procedures with a lot of complications. These are procedures that are going to affect the patient's quality of life moving forward. And so this is the final therapeutic option here, is this surgery. So the goal of drugs is to stop the patient with moderate to severe ulcerative colitis from progressing all the way down for the need for surgery. So the idea is that these drugs can help constrain healthcare costs, because it's much easier to monitor patients on colitis medicines than it is to monitor a patient that has a complete colectomy, and then there's risk of J. pouchitis and infections and things like this. It's not, colostomy bag and things like that. It's just not a great quality of life, and it can get very expensive, especially if there's complications following the surgery. So the idea is that the drugs can help prevent these patients from needing that surgery. So how are we going to stop this for the patients that are refractory to standards of care, patients that are heading towards surgery? Is that we need to develop new drugs. And so in this case, etrasimod is that new drug, and that's what we're going to study. So etrasimod targets S1P. So there's a few different S1P drugs on the market or in development, these three right here. And so etrasimod is blocking different S1P receptors. So here we have sphingosine 1 phosphate 1 receptor. We have S1P1, S1P3, S1P4, S1P5. So the different drugs have different activities on these different receptors. But the idea is that these receptors are expressed on the lymphocytes, and therefore we can block these receptors and prevent lymphocytes from exiting the lymph node. They enter the lymph node through the afferent lymphatics or the high endothelial venules, and then they get trapped. They cannot egress, they cannot leave through the efferent lymphatics. So they stay stuck because this S1P signaling is very important for leaving the lymph node. So in this case, S1P is a membrane-derived phospholipid, and its signaling is involved in regulation of multiple physiologic processes and is mediated through the receptors. The receptor for S1P is called S1P1 through S1P5. S1P1, again, which is the receptor, is expressed on lymphocytes, and it plays a key role in lymphocyte trafficking, and it regulates the lymphocyte egress from these lymphoid organs. And so this is a oral selective S1P1 modulator. It also has lesser activities on S1P4 and S1P5. And what this is it's in development for ulcerative colitis and will likely be approved in the coming months. So we've talked before about this drug development regulatory path. So I'm going to focus on the path for etrasimod today. The study that we're reading is etrasimod. It's going to be a randomized phase 2 trial. So we're going to look at this phase 2 trial. Phase 2 trials are a little easier to digest than the phase 1 that we discussed. But I want to go through the preclinical research, the animal research that was done. I want to go through the phase 1 data that was done. I want to go through the phase 2 data that the quiz will be on. And then I want to go through the phase 3 and the path for new drug approval. A lot of this data was just recently released. So etrasimod. So first you need to test this in animals. We're going to test this in healthy animals. So healthy animals are really interesting because we want to look at two different relationships. What makes a molecule a drug? What makes a compound a drug? Is two key aspects. One is the pharmacokinetics, so what the body does to that drug, and then the pharmacodynamics, what that drug does to the body. And so here, the pharmacokinetics and pharmacodynamics are shown in these animal studies. So in this animal, these different animals got multiple doses, different escalating doses of etrasimod. So here you can see there's all the way from 0.03 mgs per kg, milligrams per kilogram of animal, milligrams of drug per kilogram of animal, all the way up to 1 milligram per kilogram of animal. And then these are the kind of linear curve fits that you see here. And so in this, you can see that there's a nice dose-dependent exposure. So the more drug I give to the animal, if I look at the blood over time, so this is the etrasimod concentration in nanograms per mil of blood, animal blood, and in this case, it's going to be animal plasma or sera. What I see is that there's a nice dose-dependent relationship where the bigger the dose I get, the more drug is on board in the plasma of the animal. So this drug has a nice pharmacokinetic profile where it's absorbed through the gut, and then it's eliminated. This could be, hepatic or, renal secretion. It could be hepatic metabolism. It doesn't really matter for the purposes of this quiz, but we can look at, there's an absorption phase, a maximal concentration, and then an elimination phase. So what did this drug do to the pharmacodynamics? So pharmacodynamics, how does this drug work and what does this drug do to the body? This drug traps immune cells in the tissues, in the lymph nodes, so it prevents them from getting into the bloodstream. So you can assume that the more drug we give, the less blood cells will be in the circulating blood. And this is exactly what we see. So this is the pharmacodynamic effect. This is how I know my drug is doing something. It's very important in clinical pharmacy is understanding, does the drug do something, and does it do something in a drug-like manner? What do I mean, does it do something in a drug-like manner? Here I'm talking about a pharmacokinetic and a pharmacodynamic relationship. So the higher the amount of drug on board, the better the pharmacodynamic effect you should see. And so here in this pink line and the black squares, you see strong pharmacodynamic effect. At the highest dose, you see there is very few lymphocytes, white blood cells in the peripheral blood. So by giving this drug, I've shut down the amount of immune cells in the blood. Thus, the drug works, and it does it in a dose-dependent manner. So we see here in the lowest dose in the white circles, then you increase the dose to the black circles and the white squares, and then now the black squares is I'm getting a dose-dependent suppression of circulating lymphocytes, so lymphopenia, so less lymphocytes in the tissue or in the bloodstream, and that's shutting this down. Why do I get this dip down here at 20 hours? The dip down here is circadian regulation. As we go to sleep, our lymphocytes go down because they don't need to be actively patrolling as much as we're, kind of repairing and gearing up for the next day. But you can see in the next morning, they peak again. So after I give that dose, I give, a nice dose-dependent suppression. All of these go down via normal circadian rhythms, and then they come back up the next day, but they don't come up with the highest dose. And the reason they don't come up with the highest dose is because you still have high amounts of drug in the system 30 hours later. So with this, I know 1 mg per kg should work. So that's great. So 1 mg per kg works in healthy animals, but does it work in sick animals? So in order to do this, we need an ulcerative colitis animal model. So proof of concept in an ulcerative colitis animal model of disease. So you don't need to know this animal model, but this is just showing how the preclinical research informs the clinical trial design. So colitis induction in skin mice. This is a great animal model that a number of drugs work in. And so this animal model, in this case, you're taking inflammatory CD4 positive T cells, introducing them into immunocompromised mice. And what's happening is that these immunocompromised mice normally don't have any T cells. So I'm injecting pro-inflammatory T cells that have been depleted of all the regulatory cells. And so these are pro-inflammatory T cells, CD4 positive helper cells. They go into the tissues and they start reacting to stimuli. In this case, they start reacting to the colonic microflora and they start causing colitis. In this case, if I introduce the regulatory T cell depleted CD4 helper T cells from immunocompetent normal mouse, introduce them into this immunodeficient mouse, what's going to happen is that the immune system for this mouse is going to be populated by pro-inflammatory T cells that will drive colitis. And this gives me an opportunity to test my drug. And here, this is an untreated mouse. So you have normal colon epithelium. Again, you see these nice crypts. Down here in the sick mice, these crypts have totally dropped out. You get really extensive inflammation. The histology score doesn't look great. So in control mice, looks totally fine. In the vehicle mice, you get this really nasty colitis. And this nasty colitis, you see it here with this really pro-inflammatory degradation of the colonic mucosa. And fortunately, etrasimod works. Etrasimod works in a dose-dependent manner. So the 1 milligram per kilogram dose caused a small decrease, but the 3 milligram per kilogram dose caused a pretty dramatic decrease in inflammation. And they compared this to another S1P modulator that was already through clinical trials. So how does this work? Is that etrasimod reduces pro-inflammatory cytokines and it increases anti-inflammatory cytokines. So here we have our tumor necrosis factor alpha, our interleukin-6, interleukin-1. In the vehicle mice, very high levels of pro-inflammatory cytokines. But again, now we get a dose-dependent, at least for the cytokine, a dose-dependent suppression in those pro-inflammatory cytokines. Here, we also get a strong reduction in these pro-inflammatory cytokines, IL-17A, and an increase in these immunoregulatory anti-inflammatory cytokines. So etrasimod works by not only suppressing the immune cells and suppressing their ability to traffic through the body, but it also helps reduce the amount of cytokines in the tissues, and that is associated with its therapeutic benefit. So what do you do next? Next is you need to try it in healthy patients. Healthy patients. So this is going to be called a single dose escalation in healthy volunteers. Because this isn't a cancer trial, you want to initially try this in healthy patients. You want to look for pharmacokinetics and pharmacodynamics. And really, the ultimate goal is to look at safety and tolerability. So this is a phase 1, randomized, double-blind, placebo-controlled single-dose escalation study. So adult men and women, 18 to 45 years, were given the drug and then followed up. And essentially, they started dosing at 0.1 mg per kg. And then they started looking, is it safe? What is the pharmacokinetics? What is the pharmacodynamics? And so each dose was randomized. So you had 6 patients on etrasimod and 2 patients on placebo. So you start with a very small pill, and then you give bigger and bigger pills and look at the safety, tolerability, pharmacokinetics, and pharmacodynamics. So etrasimod was really well tolerated at doses less than 3 milligrams per kilogram. However, once you started getting to higher doses at 5 milligrams per kilogram, you started seeing toxicity. So this is a systemic drug. This gets into the plasma. This distributes all throughout the body. And what you see is that in the 5 milligram per kilogram, or the 5 milligram cohort, people who got kind of an intermediate sized pill, is you got first and second degree AV block. So this is cardiac toxicity. 2 subjects got asymptomatic cardiac toxicity. So of the 6 patients that got 5 milligrams of etrasimod, all of these started having heart side effects, probably unrelated to the immune system, but this drug, for whatever reason, was causing heart side effects, probably because the heart expresses some of these proteins as well. And because of that, now you're harming patients. And so they wanted to go up to 40 mg per or 40 mg dose, but they no longer could because that 5 milligram dose was the maximum tolerated dose where they started getting adverse events. So with this is the pharmacokinetic property. So here is on a linear scale, here's on a logarithmic scale. And again, this is very well behaved. And so at 0.1, 0.3, 1, 3, and 5 is that you're all getting increasing levels on the blood. Not every molecule behaves like this. Some molecules you might administer and it doesn't have this nice linear relationship between the dose and the exposure. So this is the pharmacokinetics. So that looks good. Next is the pharmacodynamics. What are you going to be measuring in these patients? And with the pharmacodynamics, what they wanted to look at is percent reduction in lymphocyte counts. So patients with decrease, let's look at this, greater than 45% decrease in the lymphocytes. So in placebo, there's no reduction in 45% of the lymphocytes. So, before the drug was administered, they took a basal level of lymphocytes, and then there was another blood draw after the drug was administered. At 0.1 milligrams, this wasn't enough to cause a reduction. 0.3 milligrams, not enough. 1 milligram, not enough. But at 3 milligrams, you started getting a significant reduction in majority of the patients. At 5 milligrams, all of these patients got a dramatic reduction in their pharmacodynamic readout, which is the lymphopenia, the amount of lymphocytes circulating in the blood. And again, that's what you expect. This is how the drug is supposed to work. So this is telling you need, this kind of critical level of drug on board in order to cause that pharmacodynamic effect. And again, that was informed by the preclinical studies. Now we've shown it in a phase 1 clinical trial. So what does this mean? It means now we know what we need to dose in ulcerative colitis patients. And this is what we're reading today. This is a phase 2 clinical trial, 156 patients. This is a much more recent study, so hopefully it'll be interesting. It's written by Bill Sanborn. He used to be, at the time, he was chief of gastroenterology at University of California, San Diego. Just a fantastic gastroenterologist, a fantastic person, just a great, great scientist. And he's just, the clinical trials that he's run has really revolutionized the field of gastroenterology, particularly inflammatory bowel diseases. And so here, let's look at this graph on this right side. This is percent of patients in clinical remission. So on placebo, so this is after the patients had received etrasimod or placebo for a certain period of time. The patients in remission, so these are all patients that came in with active disease that was not well controlled by current therapies. These patients, placebo worked, is that these 8% of patients got into spontaneous remission. Low dose of etrasimod caused a little bump, but the high dose of etrasimod, 33% of patients were now in remission. So this is dramatic efficacy for a large portion of the patients that were enrolled in the trial. And this gives us confidence, is that we saw there was a pharmacodynamic effect. Now we're seeing that it actually translates into clinical efficacy that's meaningful for patients. So how does this work? Is that there's an induction phase. So in an induction phase, what that means is that these patients are randomly assigned to 1 milligram etrasimod, 2 milligrams etrasimod, or placebo. And they're monitored for 12 days. Is that these patients have a 28-day screening period where they are just really rough with their active disease that can't be tapped down by existing therapies. Here, they have a 12-week treatment period. And at the end of the study is they want to see how many patients still have active disease. And then from there, what they do is they exit into this extension study. So this is the induction to try to induce these patients into remission. And then there's an exit transition to extension study, where essentially what you're doing is you're taking all the patients and putting them all on the drug and seeing for all these patients, if you keep them on the drug longer, does it work? For the patients that responded to the drug and went into remission, do they maintain that remission? So it's a very complex clinical trial design, but that's just the beauty of working with somebody like Bill Sanborn, is just these very complex clinical trial designs. They can think it up and implement it in a way to give clinically meaningful results that can say, hey, this drug had this effect on this patient population. So here's another way to look at the clinical trial protocol. So this is the extension study. First was the phase 2 study at week 12. So this is the long-term safety and efficacy of etrasimod from the open label extension. So again, this initial induction period was here, and this is what's going to be the focus of our clinical trial that we're reading. And it worked. It worked to induce remission in about 33% of patients. From there, everybody who completed the trial, regardless of what they were on, regardless of what happened next, if they went into remission or continued to have active disease, those patients then went into this open label extension where etrasimod went on for 2 milligrams per kilo, 2 milligram pills, and this went for the duration of the year. So let's look at the pharmacodynamic readouts. So here, this is the percent change in circulating lymphocytes. And so in circulating lymphocytes, people on placebo had no reduction in circulating lymphocytes. People on the low dose of etrasimod had a reduction in circulating lymphocytes by about 0.5, down here, this mean change of about 0.5. And then in etrasimod 2 milligrams, these patients went down lower to minus about 0.7. And this was great. It showed a nice pharmacodynamic relationship that was associated with the dose that was given. And then in the open label extension, these patients that were all now put on etrasimod during this open label extension, they all had very dramatic reductions in their lymphocyte counts compared to placebo. And so patients, no matter where they were initially in treatment, they got on the high dose, and that reduced the amount of circulating lymphocytes as expected, to induce that lymphopenia. So how did it work? So this was published in 2021. This is not the focus of the paper, but this is just kind of a follow-up. So the percentage of patients that were seen. And so this is kind of overall shown here with a percent of patients. You can see there's more patients, 46 patients total that had some level of clinical response. And then etrasimod through the 20 through that initial study. So this is patients that were treated through the entire study on a high dose of etrasimod, that 2 milligram dose. And so if you see the percent of patients that had a clinical response, was quite high. Percent of patients that had clinical remission, again, was quite high. And percent of patients that had endoscopic improvement, that means it looks better via the endoscope, the colonoscope, was also quite high. So essentially, it doesn't matter if you are on etrasimod for that entire period, or if you were just switched to etrasimod at some portion of the study, is that there was, a number of these patients, about 60%, had clinical remission by the end of that 52 weeks. So again, showing that the drug works, and the longer you're on the drug, the more likely you are to have end up in clinical remission. So what happened once the study was published? The study published caused Pfizer, Pfizer, had just had the best-selling medical product in the history of medicine with their COVID-19 vaccines. Pfizer was able to take that money and reinvest it into their clinical development pipeline, bringing new medicines to market. And they were able to acquire Arena Pharmaceuticals out of San Diego for $6.7 billion to get etrasimod. And essentially the reason for that is that etrasimod was developed by Arena in San Diego, and Arena had pushed it forward through these phase 2 trials. But Arena was really good at discovering these molecules. Arena was really good at doing the initial clinical testing, working with Bill Sanborn to design these trials. But Pfizer is really good at doing large-scale clinical trials. Pfizer is really good at doing marketing and manufacturing and distribution and getting difficult things out to where they're needed quickly. And so Pfizer is reinvesting that money, $6.7 billion, into acquiring Arena and getting etrasimod. So this is the quote. It's going to further expand and develop breakthroughs to change the lives of people with immunoinflammatory diseases, said their global president and manager of inflammation and immunology. So essentially, there's a synergy where Pfizer could do this. And so why is Pfizer paying $6.7 billion? The reason Pfizer's paying that is because Pfizer knows it can charge a lot for this drug, and could probably on the order of $10,000 per patient per year for this drug. How can they get away with charging $10,000 per patient per year for this drug? The reason for that is that the patients won't see that cost. They'll see a percentage of it, but insurance is willing to pay that much money. Insurance knows how expensive ulcerative colitis patients are, especially when they're refractory to standards of care, especially when you consider complications with colectomy and all the expensive surgery and all the monitoring and complications that can happen immediately or even long-term after the surgery, is that insurance would much rather pay $10,000 per patient per year than have these ulcerative colitis patients not respond to any drugs and then have to go through these really expensive procedures and all the monitoring and costs and ICU visits that might follow with these patients. So if this drug can keep these patients out of the hospital, the insurance will gladly pay for it. And therefore, Pfizer can charge a large amount in their negotiations with insurance. Pfizer will charge money based off of the clinical trial results that you see here and how good the drug works. So phase 3 clinical trials concluded in 2022, 354 patients. And this here is a press release from Pfizer, and they're able to see in a large number of patients. The initial clinical trial was here, and ultimately they're kind of showing in this and their updated press release, it was 409 patients, so much more than the phase 2 clinical trial that we just discussed. But with placebo, about after 12 weeks, about 7% of patients maintained about 7% of patients had gone into clinical remission, and at 52 weeks. only 7% of patients had remission. Or with etrasimod, this boosted up to 27% and 32% at the end of 52 weeks. So just showing that even in large, diverse patient populations, this stuff still works. And so with this, I'll kind of lead you with just some clinical terminology. So the primary endpoint is the ultimate goal. For this case, it's going to be a lot of these, clinical remissions. It's that they have something they're going to specify is that primary endpoint for the trial. Secondary endpoints, this means that the study is not powered, but this is something that's interesting to the pharmacists, to the clinicians. What can we monitor to show that the drug's working? What can we monitor to show, different efficacy readouts that are important? So this could be key secondary endpoints, just other secondary endpoints or subgroup analyses, and then post hoc analyses and tertiary endpoints. These are typically more exploratory, just looking for things, again, that are interesting to the clinicians, but not super important as far as presenting to the FDA. So sometimes when your clinical trial fails, you'll do these post hoc analyses, try to identify was there a particular subset of patients that was responsive to my therapy, and then at that time do a whole nother trial focused on that subset of patients. But with this, I hope you enjoy the article, and good luck. Thank you.